last but not least, we have Isaac Sebenius from University of Cambridge. He's going to tell us about his new method for measuring structural similarity networks and his GWAS results.
Hi everyone, thanks so much for sticking through the last talk of the last session of the day. I really appreciate it. I'm Isaac, and I'm excited to be talking about my work on the genetics of structural similarity networks in the brain. I have no disclosures. So to get started, I wanted to give an overview as to what actually structural similarity is and why it's something that we're interested in studying, both generally and more specifically through the lens of genetics. So basically, as the name might suggest, structural similarity is basically, or MRI-derived structural similarity is basically if you have two brain regions, it's just answering the question how structurally similar they are. And you can answer that from using like any structural features that you might want to use, whether that's cortical thickness or diffusion-based measures, etc. And why we're interested in that is that this measure of similarity can map onto more fundamental neurobiological patterns. So for example, if two regions are more structurally similar to each other, they tend to be connected by more axons, they tend to share similar gene expression profiles, they tend to be part of the same cytoarchitectonic class, etc. So you can really use similarity as sort of a proxy to reflect more fundamental biological patterns. Obviously, there's many ways that one can measure similarity in the brain from MRI, and in this project we focus on MIND, or Morphometric INverse Divergence, which is a method for calculating similarity that we published last year. We chose this method for this project for both technical and neurobiological reasons. So technically, it's very statistically robust, and it also allows you to include many different features at once to kind of really get a really cohesive, integrative measure of similarity. And neurobiologically, there are basically three main reasons why we wanted to, why it sort of motivated our further GWAS analysis. First, and perhaps most importantly, we observed that at the group level, there was a very close correspondence between how similar two brain regions are, as measured by MIND, and the similarity in their gene expression. So if two regions are similar, they tend to express very similar sets of genes, which sort of gives some kind of neurobiological underpinning to, or transcriptomic underpinning to MIND in the first place. Then at the interindividual level, we found that compared to another method of structural similarity estimation, it was more heritable, which is really important for GWAS analysis, sort of, comparable to other structural features that you might want to study. And in terms of the interindividual signal it can capture, we compared its ability to predict subject age and found that it was more able to predict age than both DTI networks processed through various pipelines and another method of structural similarity estimation. So altogether, these really motivated further analysis of the genetic architecture of structural similarity networks through the lens of common variants, so GWAS. So to get a better picture of exactly what we did, the most important thing in creating these networks is actually choosing good features. And so to do that, we chose 4 features to really get an integrative view on structural similarity between regions. So 2 macrostructural, mean curvature and cortical thickness, and 2 microstructural, neurite density index and mean diffusivity, which were chosen because they're all quite heritable and they offer complementary genetic signal to sort of maximize the signal that we had. Then, for each pair of 23 different brain regions, we calculated the similarity between each pair of regions to generate a single structural similarity network for each subject in the UK Biobank, so over 30,000 participants. That led to 276 edges, which were the inputs to our GWAS analysis. So we did 276 GWASs at the edge level. The results of this are summarized in this Manhattan plot here. So obviously we have quite a lot of information, 276 summary statistics, which we basically summarized here, where each dot is showing the maximum significance of every variant across all 276 edges. Different variants had different interesting properties. So some of them, had very specific effects to edges connected to a single node. Others had more distributed effects and had sort of discordant effects at different part of the network, and so on. One of the really interesting things that we realized that we could do with this data is that because structural similarity is a relational measure of the similarity between regions, we can really use this analysis to help us understand how the genetics of similarity relates to the genetics of other types of brain connectivity, which we've been interested in for a very long time. So for example, functional connectivity. So what we did is actually the exact same analysis of 276 GWASs, but functional networks, the exact same networks. One of the challenges in functional connectivity for GWAS so far has been that it's actually not that heritable. The person who gave the wonderful presentation about EEG data had this exact result. And so as you can see here, it's like really SNP-based heritability at the edge level is not very high. Mine is sort of roughly 3 times higher. But what we can do is compare the genetics by genetic correlation analysis of doing a sort of like for like analysis of an edge in the structural similarity network to the same edge in the functional connectivity network. And what we found is on average positive genetic correlations with a large number of edges reaching statistical significance after correcting for the number of edges, which indicates that on average the genetics are shared underlying structural similarity and functional connectivity. But we can go one step further and use Mendelian randomization to sort of start to tease out some causal relationships between structural similarity and functional connectivity. And so what we found is that we did find evidence in a certain subset of edges for a causal relationship of increased structural similarity on functional connectivity, but not in the reverse. Of course, this is a lot of comparisons to be making on phenotypes that aren't necessarily that heritable, and we realized that these aren't independent GWASs that we're running. And so we wanted to kind of test whether there are sort of underlying genetic latent factors that can help explain a larger portion of the genetic variance, and to sort of help perhaps crystallize this result. So what we did is we compiled an edge-by-edge genetic similarity matrix. We're basically on the left-hand side over there. Each element represents the genetic similarity between each pair of edges in the network, which we found could be well characterized by 2 gradients. So the first 2 gradients of the structural similarity genetics. Where those gradients fell in the network are shown on those network plots there. We then ran 2 more GWASs at the gradient level and found that the relationship between structural similarity and functional connectivity was very much localized to this first principal gradient of structural similarity. So really, when we found that there's much more strong both correlational and causal relationships along this axis. So great, we have sort of 2 gradients with an interesting structure to function relationship going on. But what do these gradients actually mean? What do they represent in the brain, and are there other theoretical frameworks that we can help contextualize them? So in order to answer this question, we were inspired by some existing work in the literature, which had kind of a similar approach of calculating genetic gradients, but just using cortical thickness. So not an integrative approach or a network-based approach, but just using cortical thickness, and found that one of the genetic gradients related closely to gradients of distance from the paleocortex or the archicortex, which, if you're familiar with dual origin theory, are sort of the 2 origins that are predicted via dual origin theory. So for those who aren't familiar, dual origin theory is basically the idea that the brain evolved and or develops from 2 origins, the paleocortex and the archicortex. We thought that this kind of analysis, which sort of integrates lots of different structural features and explicitly models the separation between regions, the similarity, dissimilarity between regions, could perhaps build on and crystallize this result. And so to test this, we mapped our edge level gradients to the regional level in order to be able to compare the relationship between each of these 2 gradients and the gradients that might be predicted via dual origin theory. What we found is that the first gradient, G1, the one that has a strong relationship to function, is very closely aligned with a map of distance from the paleocortex, which is sort of in line with what we'd expect via dual origin theory. And to really solidify this result, we asked the question, are there any other regions in the brain that would be a more plausible origin than the paleocortex? And so we put sort of potential origin at every other region in the brain, calculated a distance map, and found that no, actually, distance from the paleocortex is the most plausible origin in this particular case for this axis. We then performed the same analysis for the second gradient and found that the most plausible origin lay in parahippocampal regions, which again aligns with sort of the second gradient of dual origin theory. And so what we have here are these two gradients of structural similarity, which align in sort of distinct ways with the 2 origins, with the 2 gradients that one would expect from dual origin theory. So to summarize, mind is a new way of studying the structural architecture of the brain via genetics. We identified 69 different loci associated with mind networks at the edge level. We identified extensive correlative and causal relationships between mind and functional connectivity at the edge level and along G1. And we found that these two gradients of mind closely recapitulated these sort of evolutionarily defined trends of dual origin theory. Thank you very much.
Any questions from the audience? I think since we're at the end of the session and we're slightly over time, we might just leave it there. And we'll thank Isaac again for a wonderful talk. Thank you, Isaac.